Children, so today we'll be discussing about Mendel's dihybrid cross. So yesterday we have discussed monohybrid cross. So dihybrid cross basically means where Mendel took two pairs of contrasting characters for study at the same time. Let's say, for example, he took plant height and seed shape for study. So in the P1 generation, he took a pure tall plant producing round seeds and he crossed it with a pure dwarf plant producing wrinkle seeds. So pure tall and pure round seeds, these were represented as capital T, capital R, capital R. Pure dwarf as small t and small r. So from here we know that when gametes will be formed, 50% of the factors will be sent over. So from capital T will come down. From capital R will come down. Similarly here small t and small r. So crossing the plants, as I said before, is actually meeting of the gametes. So in the F1 generation, it will be capital T, small t, capital R, small r. We should remember that similar alleles we have to write together. We should not be writing capital T, capital R, and small t and small r. So going by the law of dominance, this will be this is your P1 gametes and this will be F1 generation. So in F1 generation, all the plants will be tall and they will be producing round seeds. So this is what Mendel expected and that is what exactly he got, because by that time he already discovered the law of dominance. So what he did in the F1 generation, he carried out selfing. Selfing means crossing a plant with a similar plant of the same generation. So that will also have the same genotype. So from here, there will be 4 types of gametes which will be formed. Now the reason why Mendel said that 4 types of gametes will be formed, I will be telling at the end of this video. So 4 types of gametes, the combination possible will be capital T, capital R, capital T, small t, capital R, small t and small r. So capital T, capital R, capital T, small r, small t, capital R, and small t and small r. So we make a Punnett square. Write the gametes properly. Don't write the gametes somewhere else and the box somewhere else. So these are the F1 gametes. And whatever we'll be getting here, this is the F2 generation. So capital T, capital T, capital R, capital R. Capital T, capital R, small r, capital T, small t, capital R, capital R. So like that we go on filling up. The dominant allele should be written before. So these are the combinations possible. So what will be the phenotype? Let us say this will be a tall plant producing round seeds. tall plant producing round seeds, 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 tall plant producing wrinkle seeds, tall plant producing round seeds, tall plant producing wrinkle seeds, tall plant producing round seeds, tall plant producing round seeds, dwarf plant producing round seeds, dwarf plant producing round seeds, tall plant producing round seeds, tall plant producing wrinkle seeds, dwarf plant producing round seeds, and dwarf plant producing wrinkle seeds. So we are ready with it. So what are the different types of phenotype we get? So the phenotype that we are getting here are tall plant producing round seeds. So how many are there? 1, 2, 3, 4, 5, 6, 7, 8 and 9. So 9. Then we have tall plants producing wrinkle seeds. 1, 2 and 3. Then we have dwarf plant producing round seeds. 3. And at last we have dwarf plant producing wrinkle seeds. That will be 1. So we are getting 9:3:3:1. This is the dihybrid phenotypic ratio of F2 generation for a dihybrid cross. Similarly, we can write the genotype as well. Now basically what happens, when Mendel did his experiment, he found that there in the F2 generation, he got 4 types of phenotype or 4 types of plant, and in order to explain why he got 4 types of plant, he said that 4 types of gametes will be formed. If Mendel would have said that 2 types of gametes will be formed, let's say capital T, small t, I'm writing this here. If Mendel would have said that only 2 types of gametes will be formed, let's say this one, and this side also this one. So what would have been the outcome? The outcome would have been tall and round plants, in ratio 3, and dwarf and wrinkle plants in ratio 1. So we would have got an F2 phenotypic ratio 3:1. But this did not happen. What Mendel got was 4 types of plants, and in order to explain how 4 types of plant will occur, he said that 4 types of gametes will be formed. And this is called the law of independent assortment. Why independent assortment? Because according to this law, in a dihybrid cross, when the factors for one contrasting character separates, they do so independent of the factors for the other pair of contrasting character. That means, suppose capital T is coming here, capital T is not bound to come with capital R. It is independent to come with small r as well. Similarly, capital R is not bound to come with capital T. It is free to come with small t. So the factors for one pair of contrasting character, when they assort during gamete formation, they assort independently of the factors for other pair of contrasting character. So this is called the law of independent assortment. Now, so this is up to this is what Mendel did. Now we come to this phenomenon. Mendel could not discover this phenomenon. The reason is that all the factors for the 7 pairs of contrasting character which Mendel selected, the factors for all those were located in separate chromosomes, which was discovered later on. Now this process is something which is called linkage. What is linkage? in complete dominance and codominance that I will be discussing in the next video.